Polyvagal theory, a science of safety. This is printed in Frontiers in Integrative Neuroscience and published May 10th, 2022, and written by Dr. Stephen Porges. The phylogenetic journey. Feelings of safety form the foundational neural platform for sociality. Through the lens of evolution, polyvagal theory focuses on how mammals adapted many of the phylogenetical ancestral structures that evolved to support survival in a hostile world. Note that the title of the initial publication presenting the theory is a synopsis of the theory, orienting in a defensive world, mammalian modifications of our evolutionary heritage. Polyvagal theory. The title summarizes a phylogenetic narrative in which the survival of mammals was dependent on the ability to down-regulate and modify the innate defensive systems that were inherited from our reptilian ancestors. These embedded vestigial circuits with their emergent adaptive functions are embedded in the genes of mammals. For mammals whose survival is dependent on their sociality to cooperate, to connect, and to co-regulate, the ancient defense programs had to be harnessed and repurposed to enable the expression of signals of safety and calmness in proximity to other trusted mammals. Polyvagal theory's interest in investigating mammalian autonomic regulation from a phylogenetic perspective does not focus on the obvious similarities with more ancient vertebrates. Rather, it focuses on the unique modifications that enabled mammals to optimize their survival. Consistent with this theme, polyvagal theory focuses on the evolved neurocircuits that enabled mammals to downregulate the sympathetic activation that could support mobilization to fight or flee, to reduce psychological and physical distance with conspecifics, and to functionally co-regulate physiological and behavioral state. The theory focuses on the transition from reptiles to mammals and emphasizes the neural adaptations that enable cues of safety to downregulate states of defense. Within polyvagal theory, the evolutionary trend has led to a conceptualization of an emergent and uniquely mammalian social engagement system in which modified branch of the vagus is integral. Neuroatomically, this system is dependent on a brainstem area known as the ventral vagal complex. This area not only regulates the mammalian ventral cardioinhibitory vagal pathway, but also regulates the special visceral efferent pathways controlling the striated muscles of the face and head. This does not preclude other structures from being involved in mammalian social engagement behaviors or homologous structures in other vertebrates who do not share our phylogenetic history being involved in social engagement behaviors. The relationship between mothers and their nursing offspring illustrates the social engagement system in action. To survive, mammalian offspring must initially nurse as their primary mode of ingesting food. To nurse, the infant must suck, a process dependent on a brainstem circuit involving the ventral vagal complex. Survival is dependent on the infant's nervous system efficiently and effectively coordinating suck, swallow, breathe, vocalize behaviors with vagal regulation of the heart through the ventral vagal pathways originated in the nucleus ambiguous. Through maturation and socialization, this ingestive circuit provides the structural neuro platform for sociality and co-regulation as major mediators to optimize homeostatic functions leading to health, growth, and restoration. For mammals, there's a dependency between reactions to contextual cues and the functions of this circuit. Cues of threat may disrupt, while cues of safety may support or enhance functions. The sensory branches of the facial and trigeminal nerves provide major input into the ventral vagal complex. Functionally, changes in the state of this circuit through the process of dissolution will either disinhibit phylogenetically older autonomic circuits to support defense, example, predator, disease, physical injury, etc., or inform all aspects of the autonomic nervous system, including the enteric system, to optimize homeostatic function. Mammals uniquely have detached middle ear bones, which distinguish them from reptiles in the fossil record. Detached middle ear bones delineate the frequency band that enables mammals to hear species-specific vocalizations associated with social communication and provide a safe frequency band in which they could communicate without detection by larger predatory reptiles. Middle ear bones are small bones that separate from the jawbone during gestational development and form an ossal chain that connects the eardrum to the inner ear. Small muscles, regulated by special visceral efferent pathways, traveling through branches of the trigeminal and facial nerves, regulate the transfer function of the middle ear and determine the acoustic properties of the sounds transduced through middle ear structures by controlling the stiffness of the ossal chain. When the chain is stiff, the eardrum is tighter and low-frequency sounds are attenuated. When the muscles relax, the lower-frequency sound passes through the middle ear into the inner ear. In all mammalian species, based on the physics of their middle ear structures, there's a frequency band of perceptual advantage that is expressed when the middle ear muscles contract. It is within this frequency band that social communication occurs, while the low frequencies that through evolution have been associated with predators are attenuated. Interestingly, the coordination of the contraction and relaxation of these small muscles is frequently co-regulated with autonomic state, and the muscles contract when there is a strong ventral vagal tone to promote social communication and co-regulation. This coordination between listening to specific sounds and autonomic regulation provides the neurophysiological basis for sound to communicate cues of safety and trust. In contrast, when the autonomic nervous system shifts to a state of defense, the muscles relax, allowing detection of low-frequency predator sounds, which supports defense strategies with auditory cues. In this state, the acoustic perception is based towards detecting cues of threat. The link between behavioral and autonomic state and listening is obvious in the study of language delays and auditory processing problems in children. Often children with problems in auditory processing also have a behavioral state regulation limitations. This neurophysiological link identifies a potential portal to regulate the autonomic state through acoustic stimulation, which is easily observable when a mother calms her infant using prosodic vocalizations. Similarly, we can observe this potent calming influence when a pet is calmed by the voice of a human. In addition, clinicians frequently report that survivors of trauma experience an auditory hypersensitivity to background sounds and an auditory hyposensitivity to human voices. These point further support the frequently observed link between hypersensitivity and poor autonomic regulation, reflected in hyperarousal. In a recent study, our research group documented that individual differences in maternal vocalizations have differential influences on calming infants. Following an experimental manipulation known as the still face procedure, greater vocal prosody was more effective in calming behavior and reducing heart rate following the social disruption of the still face procedure. Of course, parents and pet owners are familiar with the impact of their voices on calming children and mammalian pets. Based on this link between prosodic vocalization and calming, a listening intervention known as the Safe and Sound Protocol, SSP, was developed to reduce auditory hypersensitivities, improve auditory processing, and calm the autonomic nervous system. The SSP functionally amplifies the embedded prosody in music by applying dynamic filters to pre-recorded music. Preliminary publications document the effectiveness of this strategy. The technology embedded in the SSP has received 3 patents and is marketed by Unite Integrated Listening Systems. One of the awarded claims on the patent is for the application of the technology as an acoustic vagal nerve stimulator. Through the evolution of vertebrates, there were strong trends in the structures involved in regulating autonomic function. These trends may be summarized as moving from chemical to neural, and then evolving greater specificity, efficiency, and speed through feedback circuits that relied on myelinated pathways. Evolution is a process of modification in which existing structures and circuits are modified to serve adaptive functions. In mammals, 3 primary autonomic states with specific neural circuits are observable and emerge at different times within the evolutionary history of vertebrates. In polyvagal terms, the newest is labeled the ventral vagal complex, the oldest is the dorsal vagal complex, and in between is the spinal sympathetic nervous system. Thus, evolution informs us of the sequence through which the 3 phylogenetic dependent circuits regulate autonomic function in response to survival-driven threat reactions. In humans, this sequence is replicated during gestation.